Good afternoon and welcome to the last webinar in the Sterility webinar series of 2021, delivered by Dr. Tim Sandle. Our monthly webinars have been covering all the different aspects of Annex 1 to hopefully help you prepare for when it goes live. Hopefully you found them useful. At the end of this webinar, there will be an opportunity for you to let us know what topics you would like to see in 2022. So today's webinar will cover the introduction to mycoplasma and cell product risks. Before we start, just a couple of housekeeping things to mention. If you have any technical issues, please send a short message in the questions box on the right-hand side of your screen, and we will try to resolve the issue for you. During the webinar, RSSL would like to invite you to ask as many questions as you would like, also using the questions box on the right-hand side. At the end of the webinar, time permitting, we'll try to answer as many of these as possible. Any we don't get round to, we'll answer offline and share them with you, along with a copy of the slides from today. So this is the last time this year that I'll be introducing myself. My name's Annette Russell and I'm the Sterile Manufacturing Lead at RSSL. I work with a commercial team here at RSSL and I work directly with the pharmaceutical microbiology team to support our clients who are involved in both sterile and non-sterile manufacture. RSSL can help support you on many aspects during your manufacturing process, and if you want any further information, please do contact me direct using the details you can see on your screen now. As this is the last webinar of 2021, I thought I would test your knowledge about Mycoplasma. I therefore invite you to vote for the statement which is not correct by a poll which should pop up, technology allowing, now. Right, I'm going to close the poll. So 16% of you thought that Mycoplasma were the smallest bacterial cells yet discovered. 19% thought that they lacked a cell wall. 21% thought that they are resistant to most antibiotics. The negative are not resistant to most antibiotics. And 8% thought that they were ubiquitous. So the one that's not correct is, of course, they cause human disease, which 35% of you chose. So the Mycoplasma pneumoniae is the most common infection that you might come across in human disease, and it leads to some sort of chest cold generally, but in some cases pneumonia can occur. So just a little bit of fun there. So how can RSSL support your cell and gene therapy clients? Well, we can help with characterization of your product with both bespoke bioassays as well as kit-based bioassays. And we have an extensive experience of running ELISAs, Western blots, SDS page, and immunoassays. We also have many years I've skipped ahead there. We also have many years experience in standard compendial raw material testing, but RSSL can also help with those non-compendial and often more difficult specialized raw materials that are used in this sector. We pride ourselves on our extensive method development and validation capabilities in this area. As you are now hopefully fully aware, RSSL have a microbiology team that can help with your sterility, bioburden and endotoxin analytical needs, but we can also help with some other elements of safety testing, such as extractable and leachable profiles of things like fusion bags or single-use plastics. And we can help you detect or troubleshoot any impurities that may occur in your product. Finally, RSSL can help support your facility with cleaning validation, air and water monitoring, or any of the specialised training needs you may require. This is all on top of the other aspects that we can support Annex 1 with our technical teams, examples of which you've seen many times if you've watched our webinars. Well, that's enough of me now. I'd like to take this time to introduce you to our guest speaker, Dr. Tim Sandle. I'm sure many of you have heard of Tim and subscribed to his many publications. Tim has over 25 years experience of microbiological research and biopharmaceutical processing. He is a member of several editorial boards and has written over 600 book chapters, peer-reviewed papers and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK and also finds time to be a visiting tutor at both University of Manchester and UCL, as well as helping RSSL with our webinars. So I will now hand you over to Tim.
Thank you very much. I should just share my screen. Okay, can you see the screen okay?
Yeah, we can see the backgrounds as well, Tim.
Okay, how's that?
Perfect.
Brilliant. Okay, thank you very much. Well, it's great to be back with you, say for the last of this series of webinars, and coming to the end of 2021. And we're going to be looking at mycoplasmas. And in this webinar, we'll be considering a little bit about the human pathogen risks that Annette spoke about, some of the general issues of concerns with the risks that mycoplasmas present, particularly to cell cultures and maintenance of cell cultures. And we'll also have a look at some of the bioprocessing controls, some of the good practices that can be embedded to try and reduce contamination risks, some of the remediation strategies, and then the different detection methods, principally comparing the culture-based method with the PCR method. Okay, so to begin with, what do we mean by mycoplasmas? Well, the first thing to take note of is that mycoplasma, the term mycoplasma, is both a specific reference to a genus of bacteria, but it also tends to be used as a general term to refer to a collection of members of a class of microorganisms called the monocytes. So we can have mycoplasmas and ureaplasmas and anaeroplasmas. They're all slightly different genesis of bacteria, but often we use the general term of mycoplasma. So what all these organisms have in common is that they're defined by a morphological characteristic, and that is the lack of a rigid cell wall around their cell membranes. And there's a slight dispute within microbiology whether it's a complete absence of a cell wall or whether there is something vaguely resembling, but it's nothing like the typical cell wall that you get around most of the standard bacteria. And this then also connects with the name of the class that I mentioned. So that's a derivation from the Latin word mollis, which means soft or pliable, and cutis, which means relating to the skin. An important distinction to draw as well is that mycoplasmas are permanently in this state. So they are different from L-form bacteria. So L-form bacteria also lack cell walls, and just an interesting aside, the L is named after a reference to Joseph Lister. But this is a characteristic that's acquired under a certain set of conditions. So there are specific strains of bacteria that normally have a cell wall, but under some conditions can develop into the L form, whereas the mycoplasma is always in that state. And it's in this state because they have no peptidoglycan at all. So instead they form their membranes with other components, and this also makes them stable but extremely flexible. So some general bits of information about mycoplasmas, which ties up quite neatly to the poll that Annette presented at the beginning. So they are the smallest free-living organisms. So obviously viruses are excluded from that definition. They are parasites, so they're reliant upon gaining nutrients from the host or tissues or whatever they're attached to. And some mycoplasmas can also enter the target host, from which they then use that as a location to multiply and survive for long periods of time. And they have these flexible cell membranes, as we mentioned, and they also have extremely small genomes. So mycoplasmas range from 0.58 to 0.2, and that can be contrasted with the number of bases found in E. coli, which is 2.5 times higher. They also have an extremely long generation times compared to most bacteria. So they have a very extended lag phase, which is can be up to 9 hours, and then a prolonged generation time. So this means that it takes quite a long time for colonies to form, which could then be visualized. And as we come to look at the cultural method, they can only be visualized using a microscope. So the time to form a colony is quite long. As well as being the smallest free-living organisms, are also the smallest self-replicating bacteria. And they have cell sizes down to 0.3 microns, but they can even become smaller than that under particular conditions. So that means that they can pass through a 0.45 micron filter fairly easily, and will often, if they undergo the correct changes to their cell shapes, pass through what's typically regarded as a sterilizing grade filter of 0.22 micron. So this ability to change their shape is a form of pleomorphism, and they can often form themselves into a kind of protruded funnel-like shape. So some kind of examples on the slide there, which is also an important way of getting through the filters. They're also resistant to many antibiotics, but not all antibiotics. But the reason they're resistant to common antibiotics like beta-lactams is because most of these antibiotics will target cell walls, and that same mechanism isn't there for the mycoplasmas. They're also adept at forming biofilms on solid surfaces, particularly glass or plastic surfaces. And this presents another level of resistance, particularly to disinfectant agents, and also to a variety of environmental stresses as well. So it leads to the mycoplasmas becoming relatively hardy compared to many other vegetative bacteria. They're also include among their number some human pathogens, and again Annette alluded to the most common pathogen. So the main risks to people are relating to infections of the lungs, skin, or the urinary tract. So while there's over 200 different species of bacteria that can fall under this kind of wider mycoplasma grouping, there are 18 that have been detected as human pathogens. And we have some of those listed on the slide, and Annette mentioned Mycoplasma pneumoniae as the most widespread, but there's also Mycoplasma genitalium, hominis, ureaplasma, uriculatum, and parvum as well. And in the clinical setting, particularly in the US, concern with mycoplasmas is increasing. And this is due to increased reports of mycoplasmas becoming resistant to those antibiotics that can be used against them. So given that the array of antimicrobials and antibiotics that could be used is relatively narrow anyway, the rise in resistance is causing some concern, and that's particularly around Mycoplasma pneumoniae infections, because that does represent one of the higher causes of respiratory tract infections in children and adults. Now, generally, the symptoms are quite mild. They just appear like a common cold, but in some cases they can be severe, particularly with the immunocompromised patient. And within the industry as well, mycoplasmas do present challenges. So they are an undesirable but sometimes quite common contaminant in relation to human and animal cell lines. And one of the reasons why they do present a concern is because they are difficult to detect. And the problems that mycoplasmas cause in relation to cell lines is that they can alter infected cells at the molecular level. And this can trigger visible changes in cell morphology, or it can alter growth characteristics. And given the wide importance of cell lines in biopharmaceutical manufacturing and also in research, and particularly in relation to advanced therapy medicinal products that we spoke about in the last webinar, which are emerging, then you can see the potential challenges that they're presenting. And just to continue on the point about the visualization, this is very different to other forms of more typical bacterial or fungal contamination, but it is important to emphasize that contamination of a cell culture by mycoplasmas cannot be visualized, so it won't generate the turbidity that we can often use as a detection method. It can also be that even studying the cells themselves or carrying out the typical tests on the characteristics of the cell culture is that some of the morphological changes are not immediately apparent, and some of the altered growth rates can be minimal or simply unapparent, but they can still cause the cell culture to lose whichever property or whatever test that's involved with. So there's an additional concern there. And if you take a look at different figures, then the proportion of cell cultures that are contaminated globally does vary. And I guess it varies according to regulated versus non-regulated industries, and also to the degree of controls in place. But looking across several sources, the range appears to be between 15 and 35% of all continuous cell cultures worldwide are said to be contaminated. And the challenge is this sort of approximate 200 species of mycoplasma that have been identified. And that's since 1956. So although mycoplasmas were suspected in the late 19th century, it wasn't until the mid-1950s that the mycoplasma was first described and its kind of distinct status in terms of slightly different form of bacteria was established. It also stands that the most common causes of cell culture contamination essentially boils down to about 6 species of Mycoplasma. So there's just 6 different types that are responsible for 95% of all cell culture contamination. And again, that's a global figure. And I mentioned earlier about some of the problems to do with cell morphology and growth that Mycoplasmas cause to infected cultures, but there's other issues as well. So if you remember back to one of the early slides where we spoke about Mycoplasmas being parasitic, well, this parasitic nature where they're competing for nutrients is also a factor that hinders cell growth and proliferation. Through their metabolic action, then they can also expose cells to unwanted metabolites. They can alter the levels of protein or interfere with RNA or DNA synthesis. They can also change gene expression, cell signaling, and cell morphology. Damage can be caused when if the mycoplasma is invasive to cell membranes and cell organelles. And there's also an association with sometimes triggering mutations or chromosomal changes as well in the cells. So you get a feel of the extent of the disruption that the mycoplasmas can cause. And it's worth just considering also the implications in relation to cell-based medicinal products and ATMPs. And I know this was a subject of the last webinar, but as it is a cutting-edge area and there's a great deal of investment going into it, then mycoplasma contamination is something to be aware of, particularly as human cell-based medicinal products are heterogeneous and there's a wide range of origins and types of cells. Homogeneous cells or terminally differentiated cells. We might be deriving cells that are autologous, that's originated from the same patient, or allogenic, which is from a person other than the patient. Some cells are also genetically modified, and also the investment going into ATMPs, somatic cell therapy, medicinal products, tissue engineered products, gene therapy, and so on. There's a lot of potential for cell lines to become contaminated. And it's also important in general to ensure that we are looking after our cell lines anyway. So we need to ensure that cells used in the process have been assessed for viability and integrity, that the detection of any possible changes in cellular nature that might influence the intended function are tracked, that we're assessing viability through laboratory assays, and we're able to demonstrate that the cytoplasmic membrane integrity and activity are suitable, and we might use cell staining or viability dyes or manual or automated processes, use of light microscopy or flow cytometry of a cell suspension to look at this. And it's very important that we protect the cell bank from viruses, other cell lines, or cell strains. So there's a lot of work involved in the upkeep of cell lines in general. But the topic we're interested in are mycoplasmas. And mycoplasmas, if an infection does get in, then they can spread vastly within the cell culture, and also there's a big risk to other cell lines within the same facility. And with cell cultures, mycoplasmas present a risk by remaining attached to the surfaces of cells. And we mentioned before, some can even enter host cells. And this has been demonstrated by ultrastructural studies and drawing upon immunochemistry as well as electron microscopy. And this kind of cell relationship is quite important. So again, if we go back to the parasitic nature of mycoplasmas, the relationship with the cell is important because mycoplasmas are unable to perform many of the functions that most other bacteria can perform. So they are sort of invariably dependent upon their host for nutrients. This is one of the key cell culture risks. And in some cases also, some species of Mycoplasma will actually kill the cells outright, so cytopathic in nature, and others will cause the alterations to the metabolisms or morphology and leading to the ruination of the cell line. So there's two different types of risks that need to be contended with. And mycoplasmas have developed various genetic systems that enable their attachment to host tissue. So they're highly adapted for that. And they also have highly plastic and variable surface proteins. So this is a factor of the versatility of their surface coat and size variation. And it also provides mycoplasmas the same kind of coat protein material, also helps them to avoid the immune system when they're being considered as infectious agents as well. So their virulence factor is very high, which again explains once they've infected a cell culture, why they become very difficult to remove. And in terms of how the contamination arises and with kind of consideration of the sources of contamination, well, these are quite varied and they will include people and materials and consumables, equipment, poor quality culture media itself, and the reagents used for cell culture. And that's because they are ubiquitous and are found in many, many niches. And an example of how easy they are to spread could be the result of something like the ease of droplet generation during the handling of cell cultures, or even the fact that some commercial cell cultures are actually even received contaminated in the first place. And although they have particular conditions required for reproduction, mycoplasmas are actually quite hardy. So they do have an ability to survive for long periods in dry conditions. So this is how they can be transferred in quite easily by materials as well as being in association with people. So all of this helps to emphasize the importance of trying to minimize contamination, and particularly practicing good aseptic techniques and controlling any source that might lead to aerosol generation. And we also need reliable detection methods and to test cell cultures on a regular basis. And this can help to limit the impact that contaminants might have on the facility. And we'll look at some of the detection methods a little bit later. But in terms of general controls, it's good practice to keep incoming cultures in quarantine until they've proven to be mycoplasma free. And if we find out that a culture is contaminated, then to discard that as quickly as possible. And we can build up an understanding of reputable suppliers and to purchase cultures from companies that have a good record of providing contamination-free cultures. And we can also sometimes use antibiotics, although the use of antibiotics is not recommended due to the growing nature of resistance. Sometimes we may wish to add antibiotics to particular cell cultures in order to suppress any mycoplasmas that might be present. And also controls around people. We mentioned aseptic technique on the last slide, but washing and disinfecting the hands of people working with cell cultures is really important, as is wearing personal protective equipment, and also good practices such as restricting speaking at the bench, because of particularly the Mycoplasma pneumoniae factor with people. Cleaning up any spills immediately, using pipettes to transfer liquids rather than pouring, and having good practice where we're only handling one cell line at a time and doing a thorough disinfection of the laboratory area in between using those cell lines. And for bioprocessing itself, then there should be detailed stepwise manufacturing processes that are in place to prevent or control the contamination by viruses, bacteria, fungi, and mycoplasma. And the production techniques deployed should be standardized so that they can demonstrate that the process can effectively minimize any impurities inherent from the production process, as well as being able to prevent the introduction of contaminants external to the manufacturing process. And it's important to place a suitable emphasis upon minimizing the risk of contamination from the environment or cross-contamination from other products, and also to consider the operation and design features of the area where purification is taking place, to ensure that we've got adequate HVAC system, heating, ventilation, air conditioning, and have risk assessments for the transfer of intermediates and other substances, and also to expand that to include equipment and personnel. So that risk-based approach would be similar to that undertaken in other pharmaceutical facilities, but there would be a particular emphasis upon mycoplasmas and some of the sources of mycoplasma that we spoke about earlier. It's also useful to consider the issue I mentioned about the sterile filtration risks. So just to recap, mycoplasmas, based on their sterile size and also their ability to alter their shape flexibly, can penetrate sterilizing grade filters. So these are filters that are 0.22 micron porosity rated. So this would present a concern for the filtration of mammalian and bacterial cell culture media and sera and so on. So 0.1 micron rated filters would need to be used where mycoplasma clearance needs to be achieved. And from a GMP perspective, that would then require demonstrating by the filter manufacturer, and this would be an additional test requested aside from the standard test of sterile filter qualification. So the standard sterile filter qualification would probably be using a bacterium like Bravundomonas diminuta. For mycoplasma clearance, additional testing and filter controls would be required. We also need to be careful of things like culture media. So culture media used in media simulation trials, so for example, this could be tryptone soya broth. Quite a lot of that is contaminated with mycoplasma. And it would be hard to detect this because obviously if we're looking for bacterial contamination and we had non-sterile culture media, we would probably see all of the media go down with turbidity, but that becomes more complex when we have concern about mycoplasma. So again, it's about purchasing the media from reputable suppliers or irradiating the media beforehand. Now, where cell cultures have become contaminated, attempts can be made to remove mycoplasma without damaging the human or animal cells that form the cell line, but this is a complex and timely process, and the activity of doing so is not always effective, and there's always going to be the risk of damaging the cell line as well as targeting the mycoplasma. But methods that can be deployed would include filtration, as we just mentioned, and things like the application of ultraviolet light, or with the selective use of antibiotics. But in a lot of cases, it is impossible to remediate the cell line, and we're then faced with getting rid of the cell line. So where we do not have a reliable method to remove the mycoplasma, we need to instigate a decontamination step. So it's important for good environmental stewardship, and also to stop the release of potentially antimicrobial resistant mycoplasmas into the environment, to subject the cell line to a proven decontamination step. And which also will help with laboratory controls as well. And probably the most effective way to do that is using heat. That's because mycoplasmas have a low tolerance to heat. So decontamination using an autoclave, for example, such as running at 121 degrees for 15 minutes, would be very, very effective at destroying the mycoplasmas. The d-value, or the decimal reduction time for the mycoplasmas, is far lower than an organism like Geobacillus stereothermophilus, for example. But testing is also an important subject. And in terms of pinpointing, a good test regime would look at things like the master seed, the master seed stock, the working seed stock, and all cells used in the processing, harvesting, purification, generating the final product process. So an holistic strategy is important. So very much like the assessment of bioburden in a biopharmaceutical manufacturing process, then equally tests for mycoplasma would help to inform about the level of control in relation to that process. So in terms of test methods, so for many, many years, the standard method, and the method still described in Pharmacopoeia, is the culture-based method. So the culture-based method is the use of broth media and supplemented agar, and incubating a sample from the cell culture under the appropriate atmospheric conditions. And the big limitation around the culture-based approach is the long growth times required, and we spoke earlier about the slow rate of growth of mycoplasmas. And with any culture-based microbial test, we're also faced with the dilemma around uncultivable species, which could be natural or acquired. It also stands that the culture media used for mycoplasmas needs to be specialized, and it's what's referred to as a complex culture media. So it's more exacting in exactly defining the ingredients and with the manufacturing process compared with a general medium such as TSA, for example. So the typical mycoplasma culture media would consist of heart infusion, peptone, yeast extract, various salts, glucose, potentially arginine, and a horse or a calf serum, and the serum would make up potentially up to 20% of the medium. And then to prevent the overgrowth of any other bacteria, because the media will encourage all bacteria to grow, often antibiotics to which the mycoplasmas are resistant to, but most other bacteria are not, would be added to the medium. So something like penicillium or thallium acetate could be added to inhibit the growth of other organisms. And the complexity of the medium can lead to variability in the manufacturing process. So it's important that a rigorous quality control scheme is in place to verify the suitability of the media before it's used. And the typical testing approach would be to attempt to grow mycoplasma in broth and then transfer a proportion of the broth onto agar and then to incubate the plates. And again, as I mentioned earlier, mycoplasma growth in terms of colony formation is microscopic. So this contrasts to the macroscopic cultures that we would typically see from other bacteria following a suitable incubation time. With mycoplasmas, the typical colony sizes are around 100 to 400 microns, and the colonies have a kind of distinctive formation that many people say resembles a fried egg type appearance. And the colonies are also typically embedded beneath the surface of the agar. And to achieve growth, then the typical atmospheric conditions are what's referred to as microaerophilic. So that's largely nitrogen with up to 10% carbon dioxide. And the optimal incubation time, temperature rather, is 35 to 38 degrees Celsius. And the incubation time is between 14 and 21 days. So you get a feel for the limitations of the cultural technique. With some non-cultural methods, then things like immunofluorescence can serve as a diagnostic tool to help with the identification of mycoplasmas in situ. And this involves DNA binding of fluorochromes and antibodies. But the best alternative non-culture based method is PCR. We'll have a look at PCR in a few minutes. It is important, as I mentioned before, about the testing of the culture media to subject that media to rigorous quality control testing. And again, this links back to the variability of the media and its complexity. So there's some example of mycoplasma that can be used as controls on the slide, and these are the ones that are recommended by the European Medicines Agency guidance for mycoplasma testing. And whether those ones are selected or alternative ones, then it's important that there is a range and it reflects some species that are known to be antibiotic sensitive. And this is necessary so we can detect any inhibition of mycoplasma growth in the actual test. I want to get ones that are fastidious, that have different growth rates, that perhaps have the likelihood of being a contaminant of the cell culture, and also ones that are known to be pathogenic to the cell lines under review. So it's quite important that each new lot or batch of medium is tested for growth promotion and that the challenge is low, and by low that's generally taken to be less than 100 colony forming units. If validation isn't successful or growth promotion isn't successful, then it's important that any substances that might be inhibitory are neutralized. So these can be countered through passages, or using a larger volume of medium or dilution techniques. However that's approached, the effectiveness of the neutralization process needs to be confirmed by repeating the tests, so we can show that there's no continuation of inhibitory substance after neutralization has taken place. In conjunction with the culture growth method is staining, and particularly prior to the adoption of PCR, staining was a very common adjunct to the culture-based method. And this is a form of DNA fluorochrome staining, of which the most common is the Hirsch stain. And this is a type of blue fluorescent dye, and it's commonly used in histology, designed for staining the nuclei of living or fixed cells and tissues. And the stain undergoes excitation by ultraviolet light. So when an ultraviolet light of around 350 nanometers is directed, then this excitation happens, and this can be visualized through the application of fluorescence microscopy. So here, samples infected with Mycoplasma are seen as fluorescent nuclei. And there also might be some extra nuclear fluorescence of Mycoplasma DNA that will kind of appear in sort of filamentous form. And for optimal results, it's important that some form of enrichment step has taken place before staining is attempted. So really you need to be having levels of around 100 colony forming units per milliliter in order to get sufficient cell density to enable successful staining to take place. So the staining technique is a little bit, sometimes a little bit fiddly technique. We should also be mindful of the regulations referring to Mycoplasmas. So for those working in regulated industries, there's references in the FDA where the cultural approaches are mentioned, which are in CFRs. There's a European Medicines Agency guide, which I mentioned earlier when we looked at the control strains. And there are two pharmacopoeia chapters, United States pharmacopoeia and European pharmacopoeia, that direct quite heavily to the culture-based method. But in the 1990s, there are advances with Mycoplasma detection and with the development of suitable PCR assays, and the PCR then largely replaced the ELISA-based alternative assays that we also mentioned earlier. So the PCR test is based on detection of the Mycoplasma genome, typically the 16S or sometimes the 23S RNA molecules of the most common species of Mycoplasma that will contaminate cell cultures. And there are commercial test kits that use extracted RNA DNA mixtures for testing. And a lot of the different commercial technologies on the market are now capable of producing sensitive, specific, and fairly rapid diagnosis of Mycoplasma contamination. And a large range, and certainly the most common Mycoplasma contaminants can be detected. So certainly for cell culture applications. For medical applications, there's still a slight limitation on every potential species. And the PCR can produce a sort of positive hybridization within around 2 to 3 hours. But it is important that the PCR assay is qualified. So again, known cultures of Mycoplasma should be used, and with those cultures sourced from an approved culture collection, such as the American type culture collection. And that test samples are challenged and run in triplicate and drawn at different time points, ideally in triplicate, and using at least 2 personnel, and then also the culture method run in parallel as part of the PCR qualification. It's also important that to understand that the method won't be deemed to be false in any shape or form, so we need to have accurate discrimination and avoid anything that might lead to false positive results. So we might have a risk for an inactivated raw material, and also negative controls as well, which could be the use of, say, uninoculated media as well. So again, that whole continuous aspect of good controls needs to be central to the PCR test as much as it is to the culture-based test as well. And PCR is also strengthened, much like the culture-based method and the staining method, by enrichment. So the greater cell density that we can produce, then the more robust the PCR assay is going to be. But as things stand, the pharmacopoeia focuses largely on the culture method, and that still stands as the definitive test. But both US and European pharmacopoeia allow for alternative methods to be used. So you will get laboratories that are running both methods or may have obtained sufficient validation data to switch to PCR and obtained agreement with the appropriate regulatory authority. Okay, so that brings the presentation to an end, and the idea of this was to introduce the topic of mycoplasmas and to describe the risks that they present to cell cultures. So we've looked at what mycoplasmas are, some of the characteristics of them, mentioned a little bit in terms of the medical implications and the human pathogens. We looked at the issues and concerns that are presented to cell cultures, the importance of looking after cell cultures and maintaining them properly. We looked at some of the bioprocessing controls, some of the remediation strategies should cultures be infected, and then we round up with looking at the common detection methods. So thank you very much for your attention so far, and I'll hand over to Annette for the Q&A.
Okay, thank you very much, Tim. Before I throw everybody else's questions at you, I've got a quick question about your validation process. How do you pick what mycoplasmas to use if there's so many of them?
I think without knowing any additional information about any specific cell lines, then the current literature, from a literature review, there's around 6 species which are accounting for about 95% of the global cell culture line. So they're probably a good set to work with, unless a particular cell line's found to have some very odd contaminant, but they'd normally tick the list from looking at the perspective of a range of different growth patterns and things.
Okay, no, that's interesting. I do have some questions that we've been asked during the talk. So from Wolfgang, I've got, how much is it of a problem are mycoplasma in isolated human tissue products, such as cancer tissues or T cells?
Again, see, this is very, there's not always a straightforward answer to these, because there are some laboratories with very, very good controls. Some are fortunate to receive tissues with low contamination rates. Others are going to be less well controlled, or they're going to receive tissues that say pass between laboratories and so on. But the overall figure, is around 20% contaminated, but within that there's going to be a range based on facilities and geographies, so it's a bit difficult to pin down. I think the general thing is that there always needs to be assumption that it could well be contaminated unless you can prove otherwise.
And he's also asked, how efficient is ciprofloxacin, sorry if I've said that wrong, in getting rid of mycoplasma contamination? Do you happen to know?
Yes, I was again. There are some papers that have been published that have very, very high success figures, and some which are sort of more moderate in their kind of claims. And there seems to be a little bit of variation in some of the research and a little bit of variation in some of the applications. And some of the factors that appear to be like with the mycoplasma density, whether the mycoplasmas are the sort that are simply affixed to cells or have penetrated cells. So there is reasonable success, but there's equally a little bit of literature that pulls it in the other direction. So unfortunately, a lot of these things, it's a case of sort of experimenting and seeing what the results are. But there is some weight in favour, yes.
Okay, and I have a question from Adeyemi, and they've said, can the PCR method be classified as a microbial ID rapid method?
Well, for Mycoplasma, it will, you'll find out, as long as it can detect the species that you're interested in. So it comes back to the question that Annette asked at the beginning. So what contaminates cell cultures? Well, it looks like there's a group of sinister 6 that are the most common. So most Mycoplasma PCR methods would pick those up, but they may not pick up other Mycoplasma. In terms of being a more general identification method, then again, it depends. It comes down to what probes and primers are used. So for screening for specific pathogens, yes, but it's never going to replace at the moment. If you're talking about non-Mycoplasma organisms, something like Morditov is going to be far more effective than trying to use PCR, but PCR is good for screening for the absence of known pathogens, be they more common bacterial strains like Salmonella, E. coli, or the main Mycoplasmas of concern.
Okay, that's brilliant. We've got time for just one very quick question. Hopefully it's a quick question from Nancy. She said, you mentioned Mycoplasma can leave a film, but they don't demonstrate turbidity. Would you not expect to see a film in growth media?
Yes, you would, but I think so. Yeah, I'm trying to understand the question. So yeah, Mycoplasma contamination in growth media could cause a film. There may also be a film there depending on the purity and clarity of the growth media, but either way it stands, you wouldn't necessarily be able to readily detect the Mycoplasma simply by visually looking at the media itself. I don't know if that's answered the question, but how about I just follow up by email?
Yeah, no problem. Because I guess it depends on how many Mycoplasmas you've got in there, doesn't it? As to whether you'd start to see it.
It does, and typically the contamination rates are fairly, they're high enough to cause a problem, but they're not.
Yeah.
Because they're so small and because of the reliant upon nutrients, you rarely get that bigger cell mass to be able to see.
Excellent. Okay, well, thank you for your time, Tim. Thank you for supporting us throughout 2021 with these webinars. To the people out there listening, thank you all for attending and taking the time out of your busy days to attend the webinars. We hope you found them interesting and useful. As I mentioned, any questions we didn't cover in the question and answer session, I'll forward them to Tim and we'll pass on the answers along with a copy of the slides, hopefully before Christmas, but as time's running out, don't be surprised if they don't come till the new year. As I mentioned also at the beginning of the webinar, we plan to do some more webinars in 2022, but in order to make sure that they're fulfilling what you require, we've put a little pop-up at the end of this webinar, so if you can let us know what topics you'd like to hear about. It doesn't have to be in the sterile arena, it can be across the board in the industry. Please just let us know. So I'd like to finally say thank you from me for your questions, for Tim's support, and I'd like to wish you all a very Merry Christmas from RSSL and the team, and we look forward to further supporting you in 2022. Have a good afternoon.
Thank you.